I am involved in nanomedicine since about 8 to 10 years, and I will present some of our data regarding experimental medicine in relation to Alzheimer's disease. I'm not sure that how many of you are in neuroscience, but I would like to point out that this is joint work with various organizations, as already mentioned: European Office of Aerospace Research Development, US Air Force Research Laboratory, Swedish Strategic Research Foundation. I am from Uppsala University, and also we are working with National Center of Toxicological Research in Arkansas. My wife is here, and we are working as a team. First of all, I have to show this, that whatever I am telling to you today is our personal view, and it has nothing to do with any organization or government that I am associated with. We are all gradually getting old, and therefore Alzheimer, I don't say that everyone will get it Alzheimer, but this is the disease of aging, and it can occur even in healthy population. So this was the first example by Dr. Alzheimer, who was from Germany, and this is her patient, and in 1902 he classified this disease in his name. So that's why we have this Alzheimer disease, where at the old age we try to forget everything. We do not remember, even we do not remember that we have eaten anything. We forget to remember our friends or names. So this is a very serious problem, and nowadays millions worldwide are affected with disease. Although the research started, as you can see, from 1902, but still we do not have any real medicine available so far. So what is happening to our brain? As you can see that this is a normal human brain, and after Alzheimer, you can see that this is completely shrank here and changed in shape. So therefore, it will affect our every vital parameters, not only memory, but also many other functions in our body. So there are two ways. Either we reduce these degeneration, or once it has occurred, we cannot cure, but probably we try to have some quality of life for these patients. As you can see here with this PET scan, that this temporal lobe of this brain is completely degenerated, and that is important for the loss of function. This is a cartoon showing, you might be aware, some of you, that in our human brain that is weighing about 2.5 kilograms, depending on healthy situations, we have more than 10 billion neurons. So these are neurons, and it is supported by even 5 to 6 times more glial cells, as well as endothelial cells. So here, there is accumulation of amyloid plaques, and that make these connections entirely different and degeneration. Another example of these plaques were highlighted, and here you can see that cells are completely damaged, and the brain structure appears to be fluid. This is a cartoon showing that these are neurons, then there are some glial cells, and this is the microvessel that regulates the central nervous system function, because we have a barrier that protects our brain from harmful substances. And here you can see that when amyloid plaques are developed, these are the glial cells, microglia, and macrophages all are affected equally. So the whole brain is affected. This is an example. There are several proteins are released in the brain, and one of the important proteins is tau protein. When it is increased, it can lead to degeneration of microtubules. Microtubules means the small tubes that connect different areas in the brain. So therefore, completely loss of function can occur. This is a cartoon showing that we normally have this amyloid protein, but in situation of disease, they are cut. and they allow to accumulate inside the cell. So that is leading to one of the prominent aspects of Alzheimer. And here an example in a human brain, that is the early senile plaques that can develop because of accumulation of amyloid protein. Well, the question is that people are focusing largely on neuron cells to survive. But as I told you that we have neurons and even much more higher glial cells and endothelial cells. So I am looking at the function of microvessels that regulates the health of the central nervous system. So this is an example that we have glial cells much more, 10 times more than neurons, and even endothelial cells 15 times more. So why we ignore these microvessels? Why we focus neurons? Of course, that comes later. And on these aspects, I did a work that blood-brain barrier can be altered in situations of stress. We still do not know that why people get Alzheimer's disease. Maybe it is reflecting long-term stress on human beings. That's why all are not developing, but only a few, if you have not genetical problems. So the point is that the blood-brain barrier is regulated by microvessels. This is one neuron. Normally, in a whole healthy situation, this is quite perfect, only allowing nutrients to pass, not larger proteins. And in all kinds of neurological diseases, this barrier is leaky, and we still have drugs that can modulate these functions. I can show you that when this capillary is leaky, it will not affect neurons, but also the glial cells. So the whole microenvironment of the brain is damaged. The first example of blood-brain barrier was found by Brightman and Rees in 1969, where lanthanum, is 12 angstrom in diameter, was shown anatomically that it is blocked by the tight junction, first tight junction. There are also the second one. And this is the basal lamina where the leak can come. We replicated this in our laboratory, and here is the lumen in rat, and the lanthanum is stopped here because this is a healthy cells, and this is a normal nerve cell. So what we believe that all kind of diseases can occur. Any kind of CNS insults will open the blood-CNS barrier, because some can call blood-spinal cord barrier and other kinds of barriers. So I just put blood-CNS barrier. And when it is leaky, we can have lots of changes in the cellular expression. It will lead to edema formation, means swelling of the brain, and altogether can lead to cell death. And this can also be true in Alzheimer as well. You know that in brain we have lots of neurochemicals, because the language of brain is 20% electrical and 70% chemical. So many neurochemicals, neurotransmitters are released, and they can affect the brain function in health and disease. I'm showing especially here that people are studying excitatory amino acids, and here you can see that in Alzheimer's disease, this glutamine and glutamate functions were altered at the synaptic level. There are lots of research going on, and obviously almost all chemicals in the brain are altered. Here, I'm showing this example that widespread significant reduction in GABA concentration was observed, because there is a balance. If we have excitatory amino acids, if they are much more higher, we have cell death. And to protect that, there is an increase of inhibitory amino acids. So here you can see that inhibitory amino acid GABA is reduced. It means the brain is more vulnerable to damaging agents. Of course, it is very difficult to read for you, but I just showed that many neurochemicals are altered in the brain depending on investigators. Some say that it is increased, some say decreased, some say no change. So the material is still controversial. When we have lots of chemicals in the brain, and any kind of disease, be it Alzheimer, be it trauma, can alter the brain function, we cannot count on a single drug that can change only one aspect of the function. So either we should use multi-drug or the drugs that have multimodal capacity. We came across to this drug called Cerebrolysin, manufactured in Austria, and this is a mixture of different kinds of neurotrophic factors, not one, but several, that can protect neurons, that can protect glia, and also the endothelial cells, and with also active peptide fragments. There are some trials of Cerebrolysin in Alzheimer's disease in the United States. This is still undergoing, and there are some encouraging results. So therefore, we try in experimental models this drug using various ways of delivery. I just show you that the Cerebrolysin has different kind of actions. This is neuronal survival, neuroprotection, neuronal plasticity, and neurogenesis. Means it can also encourage damaged neurons to recuperate and provide new connections. We did some experimental models using this Cerebrolysin, and there are certain papers published here. So what we can see that it has ability to protect brain in traumatic brain injury and also hyperthermic insults. Then the question comes, why we should use nanomedicine then, when the drugs can be used or they are being used over hundreds of years to treat patients? The point is that we have learned so far that if the same drug is given using nanodelivery, it can have much better therapeutic effects than the normal drug alone. The second point has come out that these nano-delivered drugs have long-lasting effects. With regard to central nervous system, as you know that many drugs are not penetrating because there is a barrier, so there could be a rapid penetration of nano-delivered drugs. The other point is that drugs should act when it enters into the central nervous system. But there are many endogenous enzymes or biological facts that try to degrade the drugs quickly because the human response or the biological response is that something is foreign and it should be gone or treated. So it could be less degradable. Now the important question is that what about toxicity of nanomaterials? And I can tell you that still US FDA has not given a guideline that how and which way the nanomedicine could be delivered, or they have any regulation, because these are still undergoing work. And very less in vivo experiments are done on neurotoxicity using nanomaterials. So therefore, in our laboratory, we are using both kinds of research, neurotoxicity of nanomaterials in vivo situation, not only cell cultures, and also we are studying delivery of nanomaterials using drugs. The other question is that there are several ways of nano delivery. Many persons use, depending on their own laboratory or interest, different ways of drug delivery. For example, soft nanoparticles, the drugs are incorporated into soft nanoparticles, delivered biodegradable materials, and nowadays, recently, it has come about nanowires. So the point is that if we give one drug with different kinds of mode of drug delivery, can we have different effects? The second point is that if we use different kinds of drug using one way of drug delivery, by just nano delivery, can we have better effects? So these are the questions at the moment required to address in nanomedicine field. We try to highlight some of these concerns in a book that was published in 2009 as a progress in brain research. And I introduce you my colleague. This is Dr. Ryan Tian from University of Arkansas Fayetteville, and this is Dr. Wang from the same institution. And this is my wife and I here. So this Dr. Ryan Tian has a US patent of titanium nanowires. And in Arkansas, titanium nanowires are used as a supplement together with drug in knee replacement because the healing is much faster. And there are certain technology where we can also bind our drugs to these titanium nanowires. Why titanium nanowires? Because they are, up to our knowledge, largely innocuous. And when we apply titanium nanowire for longer periods in higher dose, at least central nervous system that I am concerned with, They do not show any pathology. The other evidence came that when we use these nanowires, silicon nanowires, even stem cells, some of you might have heard, when they are grown on these silicon nanowires, these stem cells can live longer, and in the system, they are not degraded quickly. This is an example that they grow faster and they are still active. So these are the titanium nanowires that Dr. Ryan Tian has used. And we use this titanium nanowire delivery of cerebrolysin in neuropathic pain and submitted as one of our innovations in 2013 National Innovation Summit in United States, Washington, D.C. And to our surprise that the jury, depending on various U.S. government and military personnel, they understood that this is one of the top 20% technological advances in nanomedicine. So we are very encouraged. And there was also lots of spotlight interviews on cerebrolysin with the treatment, because this is a multimodal drug, not one drug, and can have different effects. Well, so we have used these titanium nanowires as a scaffold to bind drugs, and we have standard techniques done in University of Arkansas, US. As I showed you earlier, that the tau protein is increased in Alzheimer's disease, and one of the important aspects for having brain pathology. There are two ways to use Alzheimer's disease in experimental model. Either we should use knockout mice that is available, or the second, that the amyloid beta protein that supposed to be one of the main causes of Alzheimer's disease, we can infuse in healthy animals for 4 weeks. Then we can have very similar pathology in the brain. Here we have used knockout mice model, and you can see that this is the A beta. We measured A beta here in normal condition in mice, very high. When we have given cerebrolysin normal, it was able to reduce it. But when we have used nanowire cerebrolysin, it came also near normal value. So the point is that nanodelivery of cerebrolysin appears to be better. We have also tested this tau, and you can see that this is the normal situation after knockout mice model, cerebrolysin alone, and nanowired cerebrolysin delivery. So these effects appears to be much stronger when the drug is delivered using nanowire technology. Yeah. As I told you that we believe that blood-brain barrier could be one of the important aspects in all kinds of disease. How we can measure blood-brain barrier without giving any external tracer? There could be one region that in our blood there is lots of albumin and fibronectin, many proteins. They are prevented by blood-brain barrier in coming to the brain. So if there are disease or traumatic injury or even human cases, there are some experiments that in cerebrospinal fluid these proteins are increased. And here is one example in animal model. We have used albumin reactivity, and you can see that after amyloid beta peptide infusion, here is, you can see this amyloid beta, and this is cerebrolysin. It is less than this one, but nanowire cerebrolysin is the least one. Similarly, you have albumin increase in control cases where the infusion was made. Nanowire cerebrolysin has much less activity of albumin as compared to the cerebrolysin alone. But this cerebrolysin is still protecting as compared to the normal ones. Similarly, the glial cells are also activated, and here is only infusion, and here is nanowire cerebrolysin and normal cerebrolysin. These neurons, when we have given nanowire cerebrolysin, they appear to be much more healthier than even cerebrolysin alone, and these are the situations where they are very much damaged. Nowadays, the another point we must consider when we do animal experiments, all animal experiments are done on healthy animals. The point is that, are we right? Probably not. Because in human cases, when we get disease or trauma, many people have different kinds of secondary diseases like hypertension, diabetes, or some of them are using different kinds of drugs. So they can also modify the disease processes as well as the neuropharmacological response of drugs. So therefore, we have started giving the same treatment in situations of hypertension or diabetes. I do not have much time to share these data, but here you can see that if the animals were made diabetes by chemical means, as streptozotocin, and given the same ABP infusion, you see the blood-brain barrier leakage is much more higher, neuronal injury is much more higher than the ABP given in healthy animals. So, and of course, this is dose dependent, you can see where we have given 20 microgram and 10 microgram. So we must consider nowadays in drug development or drug discovery that we must consider that all humans are not normal when they get any disease. We have to consider these comorbidity factors together while considering any drug development, and nanomedicine is not an exception either. I can show you here some examples where cerebrolysin was given, and you can see that normal cerebrolysin was given 5 milliliter per kilogram. We get better result. But even 10 milliliter when given with diabetes were better but not the best. But when we have given nanowired cerebrolysin in lower doses, we have the best results after ABP infusion. These are the examples of albumin leakage here. The nanowired has the best result. Astrocytic activation is least seen here, and neuronal damages were largely absent when we have given nanowired cerebrolysin. These are the example here. You can see that blood-brain barrier breakdown, and I can tell you that hypertension is equally damaging. So when the ABP was infused in CHR, chronic hypertensive rats, the increase was much more higher, and when we have given nanowire delivery of cerebrolysin, it is significantly reduced. Now I asked you before that if the same drug is given using two different types of nanodelivery, can we have different effects or similar effects? Here this is an example. This is nanowired cerebrolysin and this is PLGA delivered cerebrolysin. It appears that nanowire delivery of cerebrolysin has some superior effects, although even PLGA delivered cerebrolysin was also equally effective. And this is the brain edema. We have the same results. Here is the cerebrolysin nanowired, and here is the cerebrolysin given by PLGA. And after hypertension, the pathological changes were higher. The same picture in neuronal injury. Still, we can see that both nano delivery of cerebrolysins are better, but nanowire delivery of cerebrolysin is much better than PLGA delivery of the same drug in same doses. And here is also the albumin. So my point is that even we deliver same drug in same dose for same disease, the mode of nano delivery may affect the final results. And here is different regions that showing increase or leakage. You can see that TiO2 delivered is better than PLGA, although this PLGA delivery was also significantly reduced as compared to the normal conditions. And here are another example here. This PLGA delivered cerebrolysin, nanowired cerebrolysin, albumin leakage is best here, and also the ABP deposit is least here. Some examples, I still do not know what are the basic mechanisms behind these, but just to show you, we have measured some amino acid neurotransmitters, excitatory and inhibitory. These two are excitatory, damaging, and these are inhibitory. I have shown percentage increase so that the data are clear to you, but we can see that in normal situations, the excitatory amino acids are increased very much, and inhibitory amino acids are down-regulated. When we have given cerebrolysin by nanowired, we can see that the balance is restored. So now we are working on other aspect. This corticotrophin releasing factor was discovered by Dr. Andrew Shelley. He is a Nobel Prize winner in 1977 of physiology and medicine. We are working with him to find out, can we stimulate or potentiate or strengthen the effect of cerebrolysin when we use another kind of peptides delivered together with cerebrolysin. And so we are using different kind of CRF fragments together with cerebrolysin, nanodelivery in different models. I do not have time to go further in this case. And this is this is Mr. Mike Beebe, he's governor of Arkansas. He just left office in January this year. He is a strong supporter of nanomedicine, and he is helping our lab in a big way. We are grateful to him. Now, some of the questions in final moments can be asked, that if we have labeled cerebrolysin either PLGA or titanium nanowire, what is the proof that it is releasing when it is given in the system? One of my students, Dr. Asia, has done, there are two ways. One is titanium nanowire, and the other we are working, the nanospheres. I cannot go beyond this because there are certain limitations and it is not approved to show the data. But you can see here that when we have given in either way, there is a gradual increase in cerebrolysin level. The data is shown up to 3 hours. We have working on this. The point is that when we have delivered cerebrolysin, there is a gradual slow increase in the system, and that can have long-lasting effects. What about PLGA-loaded cerebrolysin? Here you can see that this is 1400 minute. The cerebrolysin levels are increased, and the free cerebrolysins are also not very much. What about the storage? How can you prepare this drug and keep how long time? This was studied in the blood, and you can see here that even at 4 degree or 25 degree, even 150 days, the cerebrolysin remains with the system and not degraded. We are also working with Dr. Harry Croto. Some of you are very familiar on the aspect of this nanodelivery. And some of the cerebrolysin data were well cited also. So I can say, based on some of the evidences I shared with you, that nanodelivery of cerebrolysin has superior effects than the parent drug. Hypertension and diabetes worsen Alzheimer disease pathology, so as even traumatic brain injury. Titanium nanowire delivery of cerebrolysin appears to be slightly better and may be significant at certain stages as compared to PLGA. So therefore, the mode of nanodelivery is also important in any model, and we require more research in this area. Our research is supported by various organizations, and one of the important pillars are Ever Neuropharma, who produces cerebrolysin, Swedish Strategic Research Organization in Stockholm, and of course, Wright-Patterson Air Force Base in Ohio. And there are lots of other organizations. I listed some of them. I will not go through. It's a large collaboration at different levels. I can mention here Dr. Herbert Mosler, who is the CEO of Ever Pharma in Austria, Dr. Dafin Mureșanu, our close friend from Cluj, Romania. Ryan Tian, you have shown him, I showed you. Dr. William Slicker, he is currently the director of National Center of Toxicological Research. And I stop here to listen to you about the comments and criticism of this work so that we can go further scientifically correct way. Thank you very much for your kind attention.